all on the third day of Pharmacovigilance Week, entitled Encouraging Reporting of ADR by the Patients. As we all are aware that physicians, nurses, pharmacists are the backbone of the healthcare system who contribute for the patient safety. So, our Indian Pharmacopoeia has decided to celebrate Pharmacovigilance Week on the occasion of Patient Safety Day in order to make the public and healthcare professionals aware about the importance of adverse drug reactions, reporting to improve patient safety. Our warm greetings to Professor Bikash Medhi, Professor, Department of Pharmacology, PGI Chandigarh, Editor-in-Chief, Indian Journal of Pharmacology, founder of Experimental Pharmacology and Neurobehavioral Laboratory, Coordinator, PGI Pharmacovigilance and Materiovigilance Center, Regional Coordinator, NADA, Ministry of Youth and Sports, Government of India, co-convener, India Initiate Program, ex-secretary, Clinical Pharmacology of Indian Pharmacological Society, and sir is facilitated with prestigious awards like Dr. D.N. Prasad Memorial Award, Dr. V.K. Bhargav Award, and Dr. B.N. Ghosh Award. Our another warm welcome to Dr. Ajay Prakash, Associate Professor, Department of Pharmacology, PGI Chandigarh, Deputy Coordinator, Pharmacovigilance Center, PGI, Associate Editor, Indian Journal of Pharmacology, Member, Innovation Council, PGI Chandigarh. So I would like to request our today's speaker, Dr. Shreya Gupta. Ma'am is MD Pharmacology from PGI Rohtak. Ma'am, area of interest is pharmacogenomics, clinical trials, and ethics. I would like to request Shreya ma'am to discuss in brief regarding today's occasion. Please, ma'am.
Thank you for the introduction. I will just share my screen. Just a minute. I hope my screen is visible to everyone.
Yes, ma'am.
Yes, ma'am.
Okay. So good morning, everyone. I'm Dr. Shreya. I am currently pursuing DM Clinical Pharmacology from PGI Chandigarh. So today I'll be talking in brief about pharmacovigilance and materiovigilance. What is the concept and what are the various tools and applications of the same. So I'll be discussing the concept and history of pharmacovigilance, adverse drug reactions, its importance, the setup of pharmacovigilance, the various roles and responsibilities that various people have in this setup, then what are the communication, what is the chain of communication we follow, what are the various digital tools for collection and analysis of the ADR reports, then a brief about materiovigilance, its concept, and what are we looking for in the future, India's journey so far and the way forward. So pharmacovigilance is basically the science and the activities which relate to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problems. So basically what do we do in this is that we aim to collect, to monitor, to do research, to assess, and to evaluate the information that we are getting from various healthcare providers and the patients, because all the patients in the hospital, they are consuming some or the other medicine, right? They are consuming someone is getting blood transfusion, someone is taking herbal medicines or vaccines, or someone is on, for example, some stent or some other medical device, correct? Or even some traditional and complementary medicines, that is the Ayush side of the therapeutics. So a lot of patients are consuming variety of drugs, and some of them are consuming more than one kind of drug at a time, right? So pharmacovigilance basically helps to monitor and assess whether there's any adverse reaction or any side effect which is known or unknown that any patient is experiencing from the use of any of these products. So basically the aim is to identify such events and to further analyze them so that we can prevent harm to patients. For example, if there is a product, we know it has some certain side effects, so we can add a box warning on the label, or we can avoid use in certain set of patients. So that way, pharmacovigilance is a backbone wherein we can help to prevent harm to patients due to any use of any medicine or device. So if we look at a brief history, that is from where this concept actually evolved. So it dates back to, certain side effects that were reported in the early days when the use of medicines were not well regulated. There were no, real clinical trials which used to happen, and there was no, systematic reporting of adverse events. But there used to be certain incidents which actually led to the awareness that this is something that we have to do on a regular and on a consistent basis. So in 1948, in that time, so chloroform used to be used for anesthesia, right? But chloroform right now, it is not advised anywhere. Even animal ethics have banned chloroform. So basically, this was used as an anesthetic agent, and it led to the death of a 15-year-old girl. So this created a lot of hue and cry in the UK and in the US that chloroform led to the death. Then in 1937, there was an elixir called sulfanilamide. So basically, this is the image, if you can see, the glycol, diethylene glycol added to sulfanilamide till 13. So this is the event that had happened back then in 1937. So this used to be used as an antiseptic, and it led to the death of many people. So then further in 1938, the US FDA was established, and the US had, they resolved to sort of renovate their healthcare system and to include a regulatory body for the drugs and the various food products that are being used. Okay. Then in 1961, a very famous tragedy, which I'm sure you all must be aware of, is the thalidomide tragedy, right? So thalidomide was a drug which used to be given in pregnant females for prevention of nausea, vomiting. Like in the initial first trimester, you have morning sickness, right? So for that, thalidomide used to be prescribed very commonly in the West. So they realized that thalidomide actually causes phocomelia, that is seal limbs. So you can see like all of these. So this, all of this part, these are all seal limbs. So this is what they realized that thalidomide is doing. Okay, so this drug was then eventually banned, and its use was discontinued worldwide when they realized that this is the kind of adverse effect that it is showing. So the yellow card system was initially introduced in UK in 1964. So yellow card is nothing, but it was also their version of pharmacovigilance. So they had called it the yellow card. That is terminology is still being used. So basically, it is the reporting of any such incidents in a systematic manner. So in the UK, they called it the yellow card. And then further from UK, the European legislation developed, and the EMA, that is the European Medical Association, was set up. So US FDA got set up in 1938. Following that, because some tragedies were happening over Europe also. So then they established the European Medical Association, right? In the middle of this, the WHO became active, and the International Program for Drug Monitoring, that was instituted in 1968. Okay, and then this further evolved, and Udravigilance. Udravigilance is basically the European Union's version of pharmacovigilance. So that came under the EMA. And the WHO began implementing these programs in various countries all over the world. And this is how the evolution of pharmacovigilance came into being. So this I've already discussed. So if you look at it from the Indian perspective, so in back in 1986, the ADI monitoring system was proposed for India. That time we had 12 regional centers. Then in 97, India joined the WHO ADI monitoring program, and we had 3 centers in AIIMS, KEM Mumbai, and Jawaharlal Nehru Medical College. Then in 2004 to 2008, the National Pharmacovigilance Program was started, in which many more regional and peripheral centers started getting added up. And finally, in 2010, the PVPI, or the Pharmacovigilance Program of India, that began. So now it's important to understand, even from a pharmacy perspective, what is the difference between an ADR and an AE, that is an adverse event. Okay, so an ADR is basically that we know we are giving the drug to a patient, and we see a response which is noxious, unintended, and it occurs at the doses which are normally used. That is, we know that this is a kind of side effect which may occur if we are giving it to a patient at this particular dose. Okay, so it is normally being used. It is normally being seen at that particular dose. So that drug may be given for prophylaxis, diagnosis, or therapy, whatever. So we are anticipating that this is an adverse effect which can happen. So that is an adverse drug reaction. Whereas what is an adverse event? Now, an adverse event is something which is, it may or may not be related to the drug. Okay, like let's say a patient is hospitalized, he's doing okay, and all of a sudden he develops atrial fibrillation, or all of a sudden he develops seizure, and before that he was okay, and this is a new onset event which we do not know how it happened. Now, it may or may not be an adverse effect of a drug, or it may or may not be the lack of a drug, but we are not sure. So this is an adverse event which can present during the treatment with a pharmaceutical product, okay, but we do not know whether it is directly related to the drug that we were giving or not. And this is where we have to evaluate and see whether it actually had a causal relationship or not. So if you look at the various types of ADRs, now there are a lot of ADRs which we see in the hospitals, and they are basically defined in a certain manner. So we use the mnemonic, A, B, C, D, E, F. Okay, these are the mnemonics that we use to sort of remember the ADRs, what type of ADR we are seeing. Okay, so first is the common type, augmented, that is dose related. That is, we know that it is a predictable reaction to an adverse effect of a drug which is commonly seen. Okay, so now and we know that if this reaction is severe enough, then we might need to withhold the drug, or we might need to switch over to some other therapy. Okay, so for example, we know that digitoxin is very toxic. It has multiple side effects on the heart. It can lead to multiple other kinds of arrhythmias. So if we are seeing a particular type of arrhythmia which the patient is not able to tolerate, so we will switch the drug. Or if serotonin syndrome develops with SSRIs, or any other medicine concomitant usage, which is increasing the serotonin levels. So we know that this is a toxic effect seen due to this drug. So we either withhold it or we switch it, okay, to some other drug. So this is a dose-related or augmented reaction. Second is a bizarre reaction. That is, it is uncommon. It is not, related in a dose response fashion, right? And it can lead to mortality also, or it can lead to, worsening of the patient's condition. So now, for example, that penicillin is a highly, some patients can develop severe allergic reactions to it, right? So now that is an uncommon reaction. We know that it might happen, but it's not related pharmacologically to the action of the drug, or it is not dose dependent, right? Now, in which patient it can cause hypersensitivity, that also we don't know. But yes, if we are giving, and let's say it causes an anaphylactic reaction, so we have to withhold the drug. And very importantly, we have to make sure that in the future also that particular drug is not being used. Okay. So this is an example of a bizarre reaction, or like malignant hyperthermia, which we see with succinylcholine, right? So these are some of the reactions. Then third is chronic. Chronic means that when we are prolonged administration of a drug is being done, so it can lead to certain side effects which are not seen on acute dosage or on a short-term duration, but on prolonged, we know that this kind of action can happen. For example, when we give steroids. Now, steroids initially, it is an immunosuppressive action, and it will subside your fever and inflammatory reaction in the body. But long-term use, it leads to the HPA axis suppression, right? So patient can have Cushing syndrome, Cushingoid disease, and all of those issues can happen. So this is a chronic effect that we see. So again, then we have to decide whether we should continue the drug or whether we should stop it, whether we should substitute it. So that the decision has to be taken if such a reaction sets in. Okay. Then the fourth type is a delayed. Now, delayed is something which, we're not even seeing it in this generation. Like some of them can be seen in next generation. For example, teratogenesis, right? Now, dilethyl stilbestrol, this was a drug which used to be given in pregnant females, right? Now, later on we realized that, it used to be used as a sort of a contraceptive agent, but then we realized later on that it can lead to vaginal adenocarcinoma in the daughter of the female who had consumed it. She will not have any issue, but the daughter will have that issue. And that too we saw, during her teenage years. This is a very long-term cohort study in which they realized this, that diethylstilbestrol has such a delayed effect on a genetic level, right? So these are something which often we're not able to understand for each and every drug, and it takes many years to even document such reactions, and it takes long-term studies. But for some drugs we know this, so those drugs are avoided in that particular subset of population. Okay. Then the fifth kind is the end of use. End of use means what? That you are using a drug for a long time, now you stop it, now a reaction happens. Okay. So for example, people who abuse opioids, right? You know that if you suddenly stop it, then there will be a withdrawal reaction to it, right? Or if you suddenly stop an anti-epileptic agent, you can have breakthrough seizures, you can have relapse of the epilepsy, right? Or even for depression, when you use SSRIs, you never stop it immediately, you always taper the dose. Okay, because if you stop it, then patient can again go into depression, and this time it might be more severe than the last episode. Okay. Then the last type is failure. That is, you are giving a drug with the expectancy that it will have a certain effect, but instead it does not show any effect, and we might be using the correct dosage also, or we might be using the correct formulation, but it is not showing the intended effect. Sometimes it can be, yes, that you're using incorrect dosage, but sometimes it can also be because the formulation is not made correctly, or there was some spurious formulation was there, which is why the effect was not seen. So all of these things we need to document so that we can further analyze what was the cause for such a reaction or a failure or any of such events that can happen. So now there are two terms that we use when we are defining these kind of areas. One is a serious and one is a severe. Okay, now these are two different things. They are a little confusing. So you should understand what is a serious adverse effect. So serious adverse effect is basically, it is defined. These are the definition for a serious adverse effect. If any of such incident is happening, then we characterize it as serious, which is death, requirement of hospitalization. That is, the patient was an OPD patient, you were giving him a certain drug for a certain disease, and after, let's say, a week or 2 weeks of taking that drug, something happened and he had to be hospitalized. Okay, some adverse effect happened, some anaphylaxis, any delayed hypersensitivity, something happened and he had to be hospitalized. Okay, so that is requirement of hospital administration. Or a patient who was already hospitalized, but let's say you gave him some infusion, some medicine, and some reaction happened, and because of that, his original stay, which would have been probably for the next 5 days, it got extended to 2 weeks. Okay, so that would be prolongation of the existing hospital stay. Okay, or if you have something which led to disability in the patient, right? Let's say there is a drug like hydroxychloroquine, so it leads to a lot of eye disorders. It can lead to, correct? So it can lead to keratopathies and retinopathies. Bullous retinopathy can happen due to hydroxychloroquine. Now, this is a significant disability, right? The patient can't see, the retina is damaged. So a drug can lead to a significant disability. That will be a serious adverse effect. Okay, or is life-threatening. That is, you gave up certain drug, and now the patient was stable, but now his life is in, let's say it lead to some fibrillation, or it lead to some other ventricular fibrillation, atrial fibrillation. So that is a life-threatening condition. Or let's say you gave OCPs to a woman, and she's a smoker, and now she has a clot formation in her veins, and now that's a life-threatening condition, because it can clog her venous return in the heart or in the brain. It can lead to a stroke, right? It can lead to pulmonary embolism. So this is a life-threatening condition. So all of these will come under a serious adverse effect. Okay, whereas what is severe? Now severe is basically the intensity. Okay, now fever also can be mild, moderate, or severe. Okay, up to 99.9 you will call it mild. 100 to 102, maybe you'll call it, moderate, then beyond that severe. So any adverse effect that is happening in any patient, all of them can be categorized into mild, moderate, and severe. Okay. So when we talk about severe, we're talking about the intensity. Okay. A skin reaction can be either of the three. Okay. But it might not be serious. Even a severe skin reaction might not be serious, right? You might not require hospitalization, or it might not be disabling for the patient, but it is a severe reaction as per its intensity. Okay, so this is how we differentiate between a serious and a severe reaction. Now, there is one commonly used term in ADR reporting, which is signal. Okay, so what is a signal? So basically, when we're talking about these reports, so we get them from various patients, and now our main aim is to see, was there a relationship between the administration of the drug and the event that happened, right? So what we try to establish is a possible causal relation. Okay, so that is what is a signal. Now if for example we see that yes, this certain drug is leading to this particular adverse reaction, and not one but multiple reports are suggesting that there is a reaction that is happening, so we are establishing a possible causal relationship, right? So this would constitute a signal. But usually we don't just do it on the basis of one report. So we'll have multiple reports, and we'll see the seriousness, we'll see the severity, right? We'll see where are we getting this information from? Is it a reliable source of information, right? Or is it being randomly reported? So all of these things we evaluate, and then we see if there is a signal that is being generated for a particular drug. So this is how the flow of signal generation happens. So there are certain events which are serious or which are severe enough to alert the authorities that okay, this particular drug might be leading to something which we did not know before. Okay, so EHR stands for the electronic health records, right? So in the various databases we check that okay, is this kind of a reaction being reported from elsewhere as well. So with that, we are doing the data extraction, then signal generation happens, right? Now, it's important for us to see the evidence. As doctors and as medical professionals, we have to substantiate whatever we are being, whatever we observe. So we look at the literature, we look that were there any case reports that have been published, okay? Then we see what is the actual known side effect profile? Is this a part of even the uncommon reaction or not? Or is this a completely new entity that we are seeing? Okay. Then we will try to analyze the pathway. That is, how is it that this particular thing is happening with this particular agent, right? We want to see, is it because of the chemical entity per se? Or let's say there is an impurity in a certain batch which led to this? Or was there a wrongful administration? Or maybe let's say it was contraindicated in a certain group of patients, like aspirin causes Ray's syndrome in children, right? We know this. Now, if there are reports happening of hepatic failure in children who have been prescribed aspirin, now this is something which is anyways not indicated for children, right? So we would not consider this as a new event, because we know this is a wrongful administration of drug, right? So all of these things we will see. Then even some in silico simulation techniques and all can be used to understand, is there a way in which this drug, let's say we understand that it is toxic in a certain way. So can we do some modifications to make it less toxic or, more suitable for patient administration, right? So all of these things we do. And then we see how we can proceed forward. So now I said that, for a signal generation, we see the causal relationship, right? So now causal relationship also involves causality assessment, which means that we have to see whether the event that happened, the adverse event that happened, is it because of the drug? So now we need to have some scales or some methodology to ascertain that, right? So we have some scales. We have WHO UMC, which is most commonly used, and we have the Naranjo scale. So I'll discuss that, but this is the most commonly used scale, and it's also employed in our ADR reporting forms and everything. So when do we say that adverse reaction is certain? That is, we are certain that this reaction has happened because of this XYZ drug. Okay. So basically, there are two kind of tests that we can do in a patient to see. One is called a D-challenge, one is called a re-challenge. Okay. What is D-challenge? D-challenge means that you have given a drug to a patient and you are seeing that, okay, there is some skin eruptions that are happening. Let's say sulfasalazine is being given and we see, okay, he's having some sort of rash in his forearm. Okay, after a week of administration, some rash is being seen. Now you tell the patient to stop the drug. Okay, we stop it for some time and we see whether that rash that happened, is it regressing or is it still persisting or is it increasing, right? Let's say, for example, the rash regressed. Okay, so what is this? This is called a D-challenge positive. That means we were thinking that the rash may be because of sulfasalazine. We stop the drug, the rash subsides. Okay, so when we D-challenge the drug, the rash subsided. Now there's another concept which is called re-challenge. Re-challenge means that is that adverse reaction happening again when we restart the drug? Okay. Now let's say you want to confirm that is this rash really because of sulfasalazine? So you again start the drug, maybe on a lower dose, but you see that the rash is coming again, right? So you are certain, okay, this adverse reaction is because of sulfasalazine only. Because the moment we restarted, it is again coming back, right? So this is how we determine a certain causal relationship. Okay. So we know that the response to the withdrawal was also positive and to reintroduction was also positive. Okay. And also you can have it substantiated with certain lab investigations. For example, you see that, okay, now even a skin rash is an inflammatory reaction. So you gave sulfasalazine, you did some blood tests, you saw CRP levels are going up, or interleukins levels are going up. And the moment we stop the drug, they again fall down. Moment we reintroduce, again they are going up, right? So we are having an objective relationship as well that is being established with the drug. Okay, so all of this is a certain. Then we have probable or likely. Probable is that D-challenge is positive, but re-challenge is not, or it could not be done. Okay. So, for example, that, the same example, you were giving sulfasalazine, you saw a rash. Now you stop the drug, the rash disappeared. But the patient says, no, I will not take this drug again. I don't like this drug. It makes me itch a lot. I will not take it again. Okay. So what happens is that you cannot re-challenge the drug. Or there are some cases in which you should not also re-challenge, because you might have a similar reaction. So the physician also decides, no, I will not give this drug. I have to withhold it. So in these cases where D-challenge led to subsiding of a reaction, but we did not re-challenge it, that's called a probable. Okay, so that's a probable causal relationship. It is more likely to be because of that drug, because we cannot find any other reason also for it, but we were not able to re-challenge it. Okay, then possible. Now possible means that we have a reasonable time relationship with the drug intake and the reaction. But let's say a patient was taking 2-3 drugs. Okay, let's say he was taking amoxicillin, he was taking sulfasalazine and something else as well, maybe methotrexate as well, and we saw a rash developing. But now we don't know which actually caused that. It can be because of any of the drugs. So possibility exists because of other drugs as well, right? And we, as an analyzer of the reports, we don't know if there was any drug withdrawal which led to subsiding of that reaction. So we reported the patient was on these 3 drugs and this happened, but we don't know which drug was it. Maybe it subsided on its own after a while, but we're not sure. It can be because of multiple reactions also, or maybe it was some other food allergy, we don't know. But it is likely, but not maybe possible, but we are not 100% sure. Okay. Then unlikely means that the whatever abnormality that we saw, it makes the relationship improbable but not impossible. Okay, that means ki again other drugs or flare up of the disease can also lead to that, but we're not sure ki what is it, right? So similarly, unconditional, unclassified, unaccessible, unclassifiable. So these are also reports in which we require much more investigation, and the current information that we have is not sufficient to say that there is a causal relationship, right? It might or might not be, maybe because of other factors. So this is one way in which we assess causality assessment, and this is the most commonly used scale, the WHO UMC scale. Then another scale is the Naranjo scale. Okay, so they have basically a question-based approach: yes, no, don't know, right? So like, were there any previous reports? Did the reaction appear after the suspected drug was administered? Then the third question is basically a D challenge, right? That did the reaction improve when the drug was discontinued? And the fourth is the re-challenge. Did it reappear when it was re-administered, right? So these kind of questions, you can rate the patient on these as a physician, and the scoring is there. So more than 9 is definite, that yes, it was because of that particular drug. Then probable, possible, and doubtful. This is another scale which can be used. So now, why is it important to report ADRs? Because see, in many countries, especially in third world countries, developing countries, ADRs can be among the leading cause of death. The disease might not be the cause of death, but the ADRs can actually lead to mortality, right? And in India, in fact, 5% of hospital admissions in India are because of ADRs only. So if we know that this is because of that, we can avoid these, right? And a lot of ADRs now, when they are under trial in phase 1, 2, 3, so it is being seen on a very limited population, correct? It is seen under controlled settings. So many times what happens is that the ADRs are unknown to the manufacturer or the sponsor at the time that the drug enters the market. Okay, so the ADR monitoring helps us to see whether this drug has any other reaction, like especially the chronic reactions or the delayed reactions that we discussed. Now these are not something which you can determine at the time of a drug approval, but when you are seeing it in the population, you are seeing how various age groups or genders are reacting to it. Then you can establish, you can have more information on it. Okay. So now, who all can report ADRs? So basically, all healthcare professionals, clinicians, dentists, pharmacists, nursing, everyone can report ADRs. Even the non-healthcare professionals, that is the patients and all, they can also report ADRs. So we have various methods by which the patients can directly report ADRs, something that they ate and they consume and they felt like it has caused a reaction, they can also report. So you can report all kinds of suspected ADRs. They may be known, unknown, they may be serious or non-serious, they may be frequent or rare. And also we now have the methodology to report it from all kinds of pharmaceutical products, okay? Not just allopathy, but Ayurvedic, vaccines, the AEFI, the adverse events following immunization is a very important part of pharmacovigilance now. Then medical devices, which comes under Materiovigilance. So I'll be discussing that soon right now. So reactions from all kinds of pharmaceutical products can be reported. So there are various tools that are available for ADR reporting. Okay. So first and the most commonly used is the ADR reporting form, right? So this is filled by the clinician or by a PV associates or nursing staff or pharmacist or any of the medical staff. So we'll discuss the form later. And along with that, we have some simpler means also. So we have mobile application, which is available as an Android app. Then you have a helpline number, which you can call toll free. And we have a lot of ADR monitoring centers in which you have, certain rooms assigned. So you can just go and, tell verbally also to the associates what is the adverse reaction that you saw. Okay. So this is important. This is the ADR reporting form that, is filled. So it consists of various sections. All of them have their own importance. So first we have the patient information part. So we don't write the whole name. We have to, preserve the confidentiality, only write the initials. We'll write the age, gender, weight. Okay, then we give a brief history of what was the reaction that was seen. Okay, when did it start? When did it stop? Or if it's ongoing, you can leave this blank, but when did the reaction start is an important date, because that helps us in the causality assessment, whether it was at the start of the drug or not, right? So we give a brief history of what was seen, right? Then we list the various suspected medications. Like, let's say the patient was on multiple drugs. Now we don't know which is the actually offending agent. So we document all of them, okay? We see their batch number, their expiration date if it is known, when, how was it used, dose, route, right? Then when was it started, when was it stopped, what was the indication for it, what it was given, and the causality assessment as per the medical professional, what they would think is the causality assessment for that particular drug, right? Then the action taken. That is all of the drugs that you've listed here. Was any of that withdrawn? Was the dose increased or decreased? Was it changed, right? Or do you not know the status? So all of this is written. Then I said, we do a re-challenge in some patients, right? So if the re-challenge has happened, so we write whether the reaction appeared when we reintroduce the drug, or we do not know, right? Then any other concomitant medications means that you can write any CAM medications that the patient was taking, any Ayurvedic preparations. So all of that is also reported. Then this section is important because we write any relevant lab tests. In some cases, like for example, certain drugs can cause agranulocytosis. So what does that mean? That your granulocytes in the blood, their level decreases. Okay, neutrophils, they become very less. So now that might not come as a clinical finding always, but it is a lab finding, and that's very important because it can lead to further infections and deterioration later on. Okay, so that is something that we mentioned. Okay, was there derangement of any values that we saw when the drug was started? Okay, then any other medical history, any allergies. Is the patient pregnant, smoking, alcohol? Why? Because we can have drug interactions because of smoking or alcohol. We can have failure of drug also, so we have to document this part. Then I had described the seriousness of a reaction, right? We had described that there are the 5-6 major criteria how we define a seriousness. So death, life-threatening, hospitalization, or prolonged congenital anomaly, disability. If we have seen any of these, we tick that. Okay. Then we see the outcomes. Let's say the patient was under your care. Now, has the patient recovered? Is he, dead? Is he still recovering? Or we do not know? Or was there a sequel? As in, was there a, let's say, disability happened? Okay. So, is there any such prolonged effect that is still being seen? So we write all of this. And lastly, we have the reporter details. So whoever is reporting shall give his or her details so that they may be further contacted for more information or for any, let's say we could establish a causal relationship. So we will convey the same to the reporter. So this is something I told you. We have this app which can be downloaded and can be used for reporting. Now, thanks to pharmacovigilance, a lot of the toxic drugs, a lot of compounds have been withdrawn from the market, right? So some of them like amineptine and estimazole, bromphenic sodium. Then lack of efficacy may also be seen in certain compounds, right? So we might approve it, but it's not showing effect. So what is the point of having such a drug in the market? Correct. So then coxibs. Once they were approved, it was seen that they lead to a lot of cardiovascular effects. So they were again withdrawn from the market because of that, right? And a lot of the drugs have now gotten black box warnings after pharmacovigilance activities because we realize that there are certain effects which have to be highlighted if we want to continue the use of some drugs. Okay, so all of these are the impact that pharmacovigilance has. So coming to the importance, again, patient safety is most important. Then IP efficacy is your basically your investigational product efficacy. right? So then SAE, serious adverse events. We want to track any serious adverse events. We want to see, is that more prevalent in a certain population, in a certain gender, in a certain age group. So all of that helps. Then risk benefit ratio. Now, what does this mean? Sometimes we know that, let's say in pregnancy, a drug might cause a certain adverse effect, but it might be required for the mother for her well-being, right? Now, this is commonly seen in anti-epileptic agents if the mother is an epileptic. So, we have to titrate accordingly. Okay, we can, able to accept a certain amount of side effects or a risk of side effects, but, it should show equal benefit to the mother as well, right? So that is where we do a risk benefit analysis. So again, this vigilance plays an important role in that risk benefit analysis. Okay. And then further, of course, we can educate the public on how to use a drug, how not to use a drug. So all of that is the importance of pharmacovigilance. So if we talk in brief about the setup, so various people and professionals are involved at various levels. Okay. So we know that, once it is reported, then it can be reported by the physicians and the healthcare professionals. Even the pharmaceuticals can report that. So then the data is collected, it is analyzed. Okay, various pharmacovigilance centers and the drug advisory committees, they also incorporate their views on the data collection and the analysis. And then we do a risk assessment. We do, we'll analyze it further. We'll see whether it is severe enough to recall the product, that is to ban it maybe, or just giving a warning might help, right? So all of these things are then evaluated. Okay. So this is basically how, what is the working structure of the pharmacovigilance setup in India. Okay. So we have the NCC, that is a National Coordination Center of PVPI in IPC Ghaziabad. So, before that, there are various steps and zonal officers which, collect and review the ADRs. So once they are received by the ADR monitoring centers, they are, further seen by the zonal officers. So we have them in all 4 zones of the country. Then they are communicated to the NCC. Okay, they have their steering committee, working group, the review panels, and also they check the quality of the reports. They'll check the completion of the reports, all of that. And then, further, if there is a significant event or, number of events that are being seen, then they're communicated to the CDSCO headquarters. Okay. And they may also be communicated internationally to the Uppsala monitoring centers, right? And then, a more larger review or larger study of the same can be done. And the marketing authorization. So the pharmaceuticals may also be communicated. So all of this chain of communication happens once a signal is generated with substantial reports. So this is basically the same in another way. So you have the administrative bodies, the national PV centers, and the ADR monitoring centers. So currently we have AMCs, that is the monitoring centers in like 24 states and union territories, and they are increasing in number with the coming of more centers all over India. So this is the Pharmacovigilance Center under the EPL lab in PGI. So we have our own AMC center. This is the helpline and the toll-free number which can be communicated and contacted for any ADR reporting. Okay, so like I said, so the communication chain is very important. So we have the National Pharmacovigilance Center. So India is one of the WHO member countries. So then there are multiple other countries. The headquarters are in Geneva. And then further, it is communicated to the Uppsala Monitoring Center in Sweden, right? And then, analysis from the same is carried downwards and it is communicated to all the member countries. Okay, so we have several, tools also, or the digital databases, like the Vigibase, Vigilize, VigiAccess, VigiFlow. So these are basically our, databases for collection of all the safety reports. So Vigibase is a WHO global database for the safety reports. Then Vigilize is again, it's more of an overview of the Vigibase, right? So these are all the various tools that are being used by the UMC Center for the collection of data from all over the countries. Then we have various analytical methods. Now we have to see that the reports that we are getting, how robust are the reports. So we measure the quality, okay, by Vigigrade. Then duplication of the reports, that is, removed by Vigimatch. Okay, so we'll avoid duplication of the same reaction for same patient, report from other professionals. Then, Vigirank, it helps in the statistical detection method for pharmacovigilance. So all these are various analytical methods that we use. So that was basically an overview about pharmacovigilance, in which we are more, dealing with the drug entity. So coming to materiovigilance, so brief about that. So materiovigilance is basically identification, collection, reporting, and analysis of any untoward occurrence that is happening because of a medical device. Okay. So in this, we are moving away from drugs and we are talking about medical devices. Okay? So devices can be something as small as a cannula or as a syringe, to something as invasive as a stent, right? Or a shunt that we put, CSF shunt and all of that. So all of these constitute the medical devices. So this was launched in 2015, and IPC Ghaziabad is the National Coordinating Center for MVPI. And the Sri Chitra Institute, that is the NCC for MVPI. Okay. So what all classifies as medical devices? So like I said, it can be as small as a as a syringe to as big as a stent or a artificial valve or something. So basically any device or implant or apparatus, appliance, which is being used alone or in combination, right? Combination as in sometimes we have certain like the Holter monitor. Okay, so we have a software attached to it, or we have an insulin pump, which is being used digitally as well. So it will in itself release insulin into the body. So that will be in combination with the software, right? And any of these devices which are being used for diagnosis, prevention, monitoring, treatment, or alleviation of a disease or disorder, or for any injury or disability, including our prosthetics, right? They'll also come under medical devices. Then if there is for investigative procedures, right? Any physiological or anatomical process, then support or sustaining life, as in your ventilators, or any other devices, the heart-lung machine, for example, which we use during surgeries. Then anything which is used for disinfection of medical devices. Okay, then control of conception. So all of these will actually constitute a medical device which comes under materiovigilance. Okay, so this is again the chain. It is similar to the pharmacovigilance. So we have reporting that can be done from the medical professionals. Then we collate the data, we analyze it. Again, signal detection happens similarly if we are having multiple reports of a same kind of event, right? Then we, further, it is communicated higher up to the NCC, to the CDSCO, and any decision regarding the withdrawal or a box warning or any of that, we do that, and further it is communicated. So again, this is basically the list of the institutes which come under the MVPI. So we can see the Biomedical Engineering PGI Chandigarh is a part of it, and we have many other institutes as well. Okay, so we have the technical support provided by the NHSRC and the collaborating center, further communicating to IPC and CDSO. So again, the reporting methodologies are similar. We have a reporting form, we have the mobile application as well as a toll-free number. Okay, so I'll just quickly go through the form. So it is more elaborate than the pharmacovigilance form. Okay, so we have various sections. We will talk about the reporter details similar to the pharmacovigilance form. Then the device category, which category it falls under. Is it a medical device? What is the use? Like, why is it being used? Therapeutic, diagnostic, preventive, right? And is it invasive, non-invasive, single use, multiple use? So all of these device specifications. And in vitro diagnosis, okay, was it used for in vitro diagnosis? Was it a machine which was being used, okay, or any other equipment? So then further more technical details are asked in the next page. Okay, So what was the device number, model number, serial number, all of these things are also important. So all of these are also documented while reporting. Then we have the event, that what was the event that happened which led to reporting of the same. So where was the event? Was it life threatening? Was it, again, the seriousness is evaluated here. Okay, then we give a brief history in our own words of the event. Okay, then the patient information and history. So what was the patient ID and age, gender, and all of that is mentioned. Then the causality assessment again is done, whether we are certain or probable or possible that it could be because of this particular devices only. And then various manufacturer details and all. So all of these are the important sections that are filled in the form for materiovigilance. Okay, so if we just take a brief look at the India's journey so far, so we can see that the ADR reporting has gone up multifolds with each passing year, and now it is much beyond what the data of 2015 we can see here. So which is a very positive thing, that the ADR reporting in India has become more robust with passing time. So now we even have an Ayush Suraksha pharmacovigilance program, in which you can even report the adverse events that we see with Ayurvedic formulations, right? So because a lot of people use the CAM medications, so their reporting is also important because they can also constitute a significant chunk in the drug interaction. So it's important to report these as well. So this is just a brief history of how the various milestones that India has achieved so far. So in 2010, our PVPI program had started. Then in 2012, the hemovigilance program also began. Hemovigilance as in the reporting of ADRs by use of blood products, blood transfusion, plasma transfusions, all of that. So then, in 2016, when bedaquiline was introduced, and we had an active surveillance for bedaquiline, right? And then the number of AMC centers, you see, they kept on increasing with the passing on years. And then in 2015, we had the Materiovigilance program that began. So now in 18, we have shifted the focus to Ayush drugs as well, that have also come under the coverage. Right, and now in 2020, of course, we are more towards encouraging patients as well to participate, for community participation in the ADR reporting and collection. So this is how we see the way forward, which seems promising because India is actively participating in various pharmacovigilance activities. So that's all from my end. Thank you so much.
So thank you very much for giving us the opportunity. I'm Dr. Vikas Mehdi from PGI Chandigarh. So basically, I look after North Zone, and we are the Center for Training and Technical Support. So all the students who had listened to the lecture, as you can say that we did not have the program. We had a program in 1982, and it was continued for 3 years. Then we had another program from ICMR. So that also continued for 3 years. But later on, we had a meeting in All India Institute of Medical Sciences, and that's how we started this program, Pharmacovigilance Program of India. So it's a big program, and under this program, as Dr. Seah has already said, that pharmacovigilance for the pharmaceutical medicine. We have later on in 2011, we started hemovigilance program, and in 2015, we started with Materiovigilance program. So there are several programs in pipeline, like biovigilance, Pharmacovigilance for any complementary medicine. Then as you can say that when you talked about ADR, adverse drug reaction, so it is also whenever you give any medicine, there is failure to therapy. So this also considered to be ADR, like antimicrobial resistant problem. So that's the reason that it also include antimicrobial resistance program also. So we had now largest network program in India when you compare to any country. So we have more than 578 centers. And at the same time, we also expanded the Materiovigilance program. As you can say that today, earlier we used to call drug, later we call it medicine, now we call it a product. So that is why all the student, you are requested to read now, how do you say devices, medical devices particularly. So as we have a regulation since 2017, it has been implemented from 1st April 2018. So there is an amendment in 2020. So read that regulation also. So because you can take up a career in Materiovigilance also. Of course, pharmacovigilance is expanding. You can take it another program like eco-pharmacovigilance, like how drugs are affecting the environment, and also you can take it as forensic pharmacovigilance. So it is also expanding. So with this, I like to thank you, ma'am, for you have given an opportunity so that we can interact. And in future, also some of the students, they can be trained. So you are very much welcome to our centers for training of the pharmacovigilance, also for dedicated training. So with this, if you have any questions, I'll be happy to answer. Any questions from the student and faculty?
I would like to request faculty members and students for any queries and any questions, please.
Yeah, somebody, go ahead, please. You had raised a hand. You can unmute and you can ask the question.
Good morning, sir.
Good morning.
This is Professor Rajmeet from Khalsa College, Sudhar.
Yeah.
So how can we send students for the training in this pharmacovigilance program to your center?
Okay, like there are training, because IPC has been designated as a training center for Asian countries also. They regularly have a training. So you can join there also. We also regularly conduct the training. And for any dedication, like practical approach, you can also send a request. So you get an approval from the Dean office, and we can train for maybe a month or something like that. That also can be arranged, because we are basically mandate of our center is to regional training center for technical and training and technical support for North India. So that can be arranged. As career-oriented, after 2013, pharmacovigilance has a huge ground. And let me tell you that last Gazette notification was there in 2018, I think. So that Gazette notification says that any pharmaceutical company, when they market, they have to establish the pharmacovigilance, cell in a company. That means there is a lot of scope to get into the pharmacovigilance in the industry also. Any other questions? I want that students would, ask the question, and please read that also. Like all the guidelines are there in IPC, or you can also go to Uppsala Monitoring Center, that is in Sweden, which is a WHO collaborating center. So all the activity you can also look at, and look at the DigiFlow also. We use the software. So once you are familiar with one software, then it has become easier for you to, handle the other software.
Thanks a lot, sir. Thank you so much, sir. Thanks a lot, Dr. Shreya Gupta, for the wonderful and informative lecture. I would like to thank our management and our worthy principal, Dr. Satvinder Kaur, for giving us this wonderful opportunity. And once again, thanks Dr. Vikash Mehti, Dr. Ajay Prakash, Dr. Vizar for the wonderful session. Thank you, sir.
Thank you, ma'am. Thank you. Thank you, everyone. Namaskar.
Have a nice day. Thank you.